Section 8.7, Mutations. Key concept from this section is that mutations are changes in an organism's DNA. Now these changes may or may not affect the phenotype in any way, but since they are changes in the sequences of bases in DNA, that would be considered a mutation. There are two main categories of mutations, those that affect a single gene or others types that affect an entire chromosome. Again, so just the definition of mutation is a change in an organism's DNA. Mutations that occur during replication affect a single gene, just one sequence of bases on a DNA molecule that controls a gene for a particular trait. Mutations that occur during meiosis affect a group of genes or an entire chromosome. So mutations that affect a gene, one of these would be a point mutation. And this occurs when one nucleotide is substituted for another. So at one point on the DNA molecule, one base is substituted in for another one. The correct nucleotide would be replaced with an incorrect one. Most of the time, DNA polymerase is going to catch this type of mistake and it would fix it, but if not, it can change the entire protein. I remember when DNA polymerase is first adding bases, we'll get like 1 in 10,000 mistakes, and then afterwards we're getting 1 in 1 billion. Right, so this is before. DNA polymerase proofreads, and this is after, much more accurate after. Okay, but if we're getting just one incorrect base, it will be called something called a point mutation, and it can change notice it says it can change the entire protein. Types of gene mutations that are caused by substitutions. The first one would be a missense mutation. So this would be one base is being replaced for another one. UUU codes for phenylalanine. UUU, when that is on the code from DNA, that is transcribed into RNA, would code for the amino acid phenylalanine. If UUU or UUA, it would code for leucine, which is a different amino acid, and as a result it would change the shape of the protein being formed, and that'd be mutation. The next is a silent mutation. Okay, silent means it doesn't cause a change in the amino acid at all. Remember, a lot of these amino acids can be coded for by more than one codon. So if the original one was GUU, codes for valine, GUA, there's a mutation, would also code for valine. Even though these nucleotides are different, you still get the same amino acid, so it's a silent mutation. There is a mutation, but it causes no change in the gene. And the last one is a nonsense mutation, where the amino acid, the codon that codes for an amino acid is changed to a stop codon. So UGU would code for cysteine. If your mutation caused it to form the messenger RNA transcript UGA, that would be stop, which means translation would stop and the protein wouldn't finish, it would just stop right there. And it would be a non-functional or a nonsensical mutation. So let's review these point mutations. All three of these are point mutations. Remember, point mutation is substituting of one base on DNA for a different base, and it can cause these different types of point mutations. Okay, so this first one, we have ATG, this one GAA as opposed to GAC, so this base A is now switched for G. And what you get is you get the same amino acid, right? Glutamic acid, it's the same one. So since this doesn't affect the protein, this is a silent mutation. In this guy, we have ATG, right? ATG. Here, GAA and TAA. So this G is replaced with a T, and what happens is rather than getting glutamic acid, we're getting stop. So if the protein stops adding amino acids before it's completed, that is going to give you a nonsense mutation. Nonsense mutation. Because this is not going to be complete, it's not going to be enough, it's basically nonsense. And this last one, ATG, ATG, those are the same. GAA and GAC, so this A was replaced with a C, and rather than glutamic acid, we're getting asparagine, different amino acid. Okay, and since we're getting a different amino acid, the shape of the protein form from it will be different, and we're getting a missense mutation. Okay, again, so these are all three types of point mutations, right? Silent, no effect on the protein. Nonsense, it just stops translation and never completes the protein because the stop codon is inserted for an amino acid. And the last one would be a missense, where it gets a different amino acid form because of the point mutation, and the protein is either less functional, or it doesn't work as good, or not functional at all, in which case you get missense mutation. So other types of gene mutations would be something called a frame shift mutation, and this involves the insertion, meaning adding, or deletion, subtracting, of one or more nucleotides in the DNA sequence. What this does is it shifts the entire reading frame. Because it changes the reading frame, it affects the protein more than a point mutation. So this is showing the mutated DNA. The original would be TAC GGT AGT CAA and ACT. So by inserting this extra base in, what it did is it moved the reading frame over from here to here, right? So all these things are off a little bit. The original strand that would be formed from this guy would be methionine, leucine, isoleucine, serine, and again leucine. But when it does this, it moves it over and it changes the sequence of amino acids from this point on, right? Methionine is leucine, but now proline is not leucine. Serine is not isoleucine. Valine is not serine. So you're getting a much bigger effect on the protein because it's changing all of the amino acids from that point, or it could change all of them from that point on. Okay, so a very big effect when you insert or delete, but this one is an insertion. So frame shift mutations would be either deletions or insertions. In a deletion, you're removing one or more nucleotides. So in this case, this U is being removed, and what it does is it slides the reading frame. All these guys move over one. Okay, so rather than methionine, leucine, glutamic acid, valine, arginine, we're getting methionine, glycine, lysine, serine, because again, since this A is moved over, this whole reading frame changes. This second base, rather than being CUA, would be CAG, the G coming from here. So it just slides this whole reading frame over, and then that would be the next reading frame, and the next, and so on. So it would look like this, which is quite a bit different. Okay, again, all that's happening is this, in this case, this A is being removed. So what happens is we get this, we get a mistake, we get a mutation. Insertion is adding adding of one or more nucleotides. So in this case, this AUG, CUA, GAA, what's happening in this case is this guy, G, is being inserted. So we're getting an extra G in there. And again, it changes the reading frame, and it changes these amino acids, everything after this point. So these three are different than these three. Okay, again, insertion. So right between this A and G, we're adding a T, and again, it's going to change the reading frame. So the codons are being read with a different sequence of nucleotides, and as a result, getting a different sequence of amino acids, which changes the protein. So just to review what we've done so far, this first example, it's showing you sequence of messenger RNA. So we have AUG and AUG and CGA and CGA, UUA, UUA, all the same so far. UAC, UAG. So these guys are different than GG. G is GGA and AAA is just AA. So what happened here? What type of mutation was this, and what happened? Well, this is a point mutation. This is a deletion. What happened was this C was removed to form UAG, and rather than coding for tyrosine, it gave a stop codon. So this is a deletion. And since a stop codon was put in instead of an amino acid, this causes a nonsense mutation. And the second guy here, okay, AUG, AUG, CGA, CGA, UUA, and UUA, all the same so far. UUC, UUA, and then CGG. So what happened here is this A was inserted, right? Inserted. And now we're getting different amino acids. We're getting leucine and arginine as opposed to tyrosine and glycine. So when it changes amino acid, this is an insertion. Insertion. That causes what type of mutation? Is a missense mutation. Chromosomal mutations are happening during the formation of gametes, which happens through the process of meiosis when crossing over occurs. If the segment that was crossed over reattaches incorrectly, a chromosome mutation will occur. So remember, during crossing over, you're getting a chunk of chromosome, a section of the entire chromosome, not just one base, not a couple of bases, but a whole bunch of the chromosome is crossing over with its homologous chromosome. If it doesn't do that properly, then you would get what's called a chromosomal mutation. It kind of illustrates a bunch of them here. We're going to talk about these on the next couple of slides, and explain each one. Chromosomal mutations. The first step we're going to talk about is what's called a gene duplication. Remember, these are happening during meiosis. So when prophase 1, when homologous chromosomes cross over, what can happen with a gene duplication is both segments of the homologous gene, the part of the chromosome that was going to cross over in it, what happens is they both reattach to the same chromosome. So the other chromosome is missing some of the gene or missing some of the chromosome. And as a result, this is called a chromosomal deletion. One side has extra, like one homologous chromosome will have extra, and one will be missing. Okay, an inversion mutation is the segment reattaches, the segment that crosses over reattaches to the same chromosome, but in reverse order, so it just flips. So it would be getting read backwards. Translocation, chromosomal mutation, would be where a piece of one chromosome moves to a non-homologous chromosome. For example, if chromosome pair number 2 was crossing over and the part that was supposed to cross over broke off and attached to chromosome 5. That's its non-homologous chromosome. So one chromosome is going to have this extra stuff, chromosome 5, and one chromosome 2 would be missing genes. Last is non-disjunction. This happens during meiosis, specifically anaphase 2 and sister chromatid separate. And what happens if the sister chromatid doesn't separate? Okay, and if it happens to all of them, which can be in plants, it's not super common, but you get something called polyploidy, where all the chromosomes fail to separate, so all of them are in one, the gametes, and none are in the other. Again, kind of rare. In humans, if it's just one chromosome, trisomy 21 is caused by non-disjunction, and this is what leads to Down syndrome. When the developing embryo fetus gets an extra copy of the 21st chromosome, because usually in the egg cell, when it's forming, the chromosome 21 doesn't separate. It sticks together, and then if that egg gets fertilized, when the sperm adds its chromosome number 21 to it, it'll get 3 copies of it rather than 2, which is the trisomy, and it causes the symptoms associated with Down syndrome. So this graphic is showing you some of these chromosomal mutations we talked about. A deletion, in this case, gene D here is being broken off and not part of this. Okay, so we just lose this gene. Okay, a duplication, where when crossing over happens and you're getting BC twice, BC and again BC, where the crossover part attaches to the same chromosome. This would be a duplication. An inversion, where these guys are supposed to cross over BCD, but when they don't and they flip and go the opposite direction, so we would have, rather than ABCDE, we have ADCBE. These guys are in reverse order, and that is called an inversion. And a reciprocal translocation, so on this one, ABCDEFGH. Okay, and on this guy, MNOPQR. And what happens is when these guys cross over, this goes to this one, and this goes to this one. Now remember, these are crossing over with non-homologous chromosomes, so this is called a translocation. Reciprocal translocation because these guys are both exchanging with the non-homologous chromosome. Mutations may or may not affect the phenotype or the expression of the trait in the organism. The effects of mutations depends on the number of genes involved and the location of the mutation. So if there's a bunch of genes involved, then you would probably expect it to have a relatively major impact. Also the location, if it happens right like in the middle of a gene or something would be important, that could cause a lot of harm, even if it coded in a section of the messenger RNA transcript that didn't necessarily code for the gene. It could be an important part where the RNA processing wouldn't recognize it and wouldn't cut out that intron, so you could get a mutation passed down that way. So the impact on phenotype, chromosomal mutations affect a lot of genes and tend to have a big effect on the organism. They can cause the gene to no longer function, or it can cause a new variation of the gene. Gene mutations occur on a smaller scale, but can also have a big impact. Many of these, however, don't affect the phenotype at all. Again, point mutation that results or causes a silent mutation that doesn't affect the protein at all, so you'd never even know that there was a mutation. There is, because it's a change in the DNA, but it doesn't affect the protein one bit. So how these mutations infect or impact offspring. Mutations can happen in both body, also called somatic cells, and germ cells. Mutations in body cells only affect the organism. They're not going to get passed on to their offspring. Mutations in germ cells can be passed to the offspring. They are a source of genetic variation, and as such will affect the phenotype of the offspring. This effect can either be a positive or a negative effect. Mutations can be caused by several different things. replication errors would be the first one, when DNA polymerase doesn't proofread the strand or isn't able to fix it, and a gene mutation occurs. Okay, this would be happening during replication. Okay, mutagens are agents in the environment that can change DNA. Certain things like radiation, chemicals, or certain infectious agents can cause the DNA to change. They can speed up replication or break DNA strands. Some are naturally occurring, ultraviolet rays from the sun, and some are man-made, like industrial chemicals, especially things called heavy metals. Those can cause some serious problems, right? Even things like cigarette smoke, and other things, even stuff that you use in everyday products. If you look at their labels, a lot of them have warning labels on them that they can cause cancer, so just be aware of that.